So you're one of ours, that you can introduce your project yourself.
Yeah, I can do that.
Linett.
I think most of you already know me, but for those that don't, I'm Linett Rasmussen. I'm a PhD student here with Tom Gilbert and Anna Johanna Svensson. And I also have a supervisor out at Copenhagen Zoo named Christina Bitsum. And today I'm going to just be focusing mainly on the background of the project, mainly due to Corona delaying the project. So I don't have a ton of results to share with you guys. And yeah, let's get started. So my project is titled A Genetic Approach to Cottontop Tamarin Conservation. And for those of you that don't know, this is a Cottontop tamarin. This is my little buddy from Bankia Zoo that I met. And these species are only found in northwestern Colombia. You can see on the map, the orange area is their entire distribution, and it's not very big. But we estimate that there's probably about 7,000 individuals in the wild, which makes them critically endangered according to the Red List. And the IUCN Red List also estimates that of those 7,000 individuals, there's only approximately 2,000 mature adults. Some of the major threats that these guys face is deforestation. So their habitat has been logged for several decades now, and there's no real sign of slowing down. This has resulted into the majority of their range being very, very fragmented and causing them to be quite isolated. These monkeys stay in the trees. If they do come down, they have a lot of predators, and they most likely will not survive if they're out in the open. Then we have also the illegal pet trade. That's a major threat for them for many, many years now. They've been viewed as cute pets to have, and that leads to a lot of capture in the wild and sold on the black market. So to give you a full understanding of why we made our project the way we did, I first want to take you back in time to explain a little about this species' history. So I'm going to take you all the way back to the groovy 60s, where there became a new threat to these guys. So in the late 60s, in a captive population, some people discovered that these monkeys could actually develop colon cancer as well as colitis. And this made them very interesting because there aren't that many species that have the same development as colon cancers as humans do, and these ones are very similar. So a bunch of medical facilities really wanted these animals so they could study them. I just realized the camera's right there and I'm standing way too close. Sorry, guys.
It's fine. It's not
Oh, okay. I was just like being only my torso. All right, so because of this interest in the biomedical research area, obviously this mass exportation happened out of Colombia and into biomedical research facilities. Because it was, the 60s in Colombia, legislation was a bit loose, not much is written down, there's not very many records. So the actual number of how many individuals were exported is unknown. There are a few records showing that about 30,000 individuals were exported to the US, and we're assuming many more were exported on top of that, as well as we know that individuals were also exported to Europe. So in total, in about a short decade, a little less than a decade, we estimate that almost up to 100,000 could have been exported along with the illegal pet trade out of Colombia in just under a decade. Luckily, a ban in 1973 was put on the exportation, and the IUCN had listed them as critically endangered. So this may be a little basic slide for a lot of you guys here, but in case there was someone that didn't know, part of our theory behind all of this is that we think that something called the bottleneck effect could possibly have happened. And that is kind of when the population decreases. So if you look at the light green shape, I guess we can kind of call it a trapezoid, you can see it's getting narrower, 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 and that is to represent that the population is decreasing. And all the circles inside is representative of the genetic diversity. So you can see there's a bunch of colors, lots of good diversity. And as we're exporting all these individuals, it's getting decreasing the population. Then we have this ban on exportation, and the individuals can start repopulating the areas. But we're lacking a lot of this genetic diversity on the other side. That's kind of the idea of the bottleneck effect. So that got us into thinking, well, what happened to all of these animals that were mass exported out of Colombia back in the late 60s, early 70s? And they were actually, after there was a ban on medical testing of these animals, they were donated to zoos all across the globe. And actually, the majority of the individuals we have now in the zoo populations are descendants from those biomedical facility animals. And that can lead into a really interesting group to study for us. And in fact, even bigger bonus, here in Europe, the European zoo region, we actually have the largest population of captive Cottontop tamarins. And the second largest is the North American zoo region. So our approach to the study was that we wanted to look at the historical population, the captive population, as well as the wild population, and compare to what, see what kind of genetic diversity we have out there. So we started off with the museum collections. We contacted a lot of museums, tried to get some samples that were from before this exportation, so before the 60s, to see what kind of diversity was happening. Then we also have gotten samples from the European zoo region, the North American zoo region, and also now the South American zoo region. And those will help us give an idea of what kind of diversity we have within our captive population. Then we have some collaborators in Universidad de Antigua, which is a university down in Colombia that is helping us sample the southern distribution of the wild Cottontops. And then we have Proyecto Titi, which is the conservation group for these guys, who is helping us sample the northern distribution. And just a quick shout out to Proyecto Titi. They're doing a lot of things to help many of the threats that these guys are facing. They're also purchasing land on top of their reserves to expand their reserves, and as well as land that can connect as corridors between different fragmented areas. So this is one of their workers that is planting a bunch of plants that will provide food as well as shelter for the Cottontop tamarins. And then they also do a lot of local education activities. And these local education activities educate the younger generation of why we would want to preserve these animals, why we want to preserve the forests, and what they can do to help, as well as they also hire locals to do all of these restoration projects and help out with them, because if they can get them an income that can replace maybe the income of them going out and grabbing an animal and selling it on the black market, that can be a huge difference right there. So then the aim of the study primarily is to look at all the diversity that we can see in the different groups, advise the breeding programs of the zoos, so that way we can preserve the highest level of genetic diversity, as well as identify individuals that could be used for supplementation if that was what we see is required of the wild population. Then we also want to be able to advise the conservation groups about how their populations look and if it looks genetically healthy or not, as well as advise on any form of translocations that need to be done. So, as you all know, usually the lab process of this is you take some sort of sample from the monkeys, usually we take hair, process in the lab, get some DNA sequence out. And as many of you also know, this is very, very expensive. And this project has a lot of samples. So shotgun sequencing, we quickly realized was going to be way too expensive for our budget. So instead, we decided to go with a method called MobiSec, which I think some of you have been hearing more and more lately. It is a cheaper technology to do genotyping. And it's pretty much a targeted library. So it's targeting the transposable elements within our genome. So if you look at the figure, here's just a quick breakdown of what happens in the lab. You start with fragmented DNA, make beautiful blunt ends like we all like to do in our beginning of our library build, add our adapters, and then you do a PCR where we have this primer that's targeting the end of the transposable element region before the TE tail. And then we get this beautiful flanking region. That's what we're interested in, is SNPs in that area. And the idea behind this is transposable elements are randomly spread out through our genome. So hopefully we'll cover a lot of the genome with able to get high coverage in those areas so we can confirm the SNPs, and as well as put a lot more samples on one lane of sequencing. So for example, I just sent off a sample where I put 165 individuals with 2 different TE primers on one lane. So a lot of information. So just a little fun diagram of what we expect to find. We'll see if we're right about this, but we think that we'll have a beautiful, diverse historical population, maybe a little less diverse captive population, but some that's still representative of the historical. And then we have the wild population that's probably lacking a lot of genetic diversity. And the idea is that we can create a constructive conservation plan along with the zoos and the conservation group to develop a future population that looks as diverse as possible. Another future direction we've been talking about, if we have enough resolution, is doing some sort of geotagging, so finding a marker that the Colombian authorities can use to identify where all their trafficked individuals that they are confiscating come from. And so hopefully we can help them out and so they can put them back into the correct forest. And that's my presentation. Thanks for listening. Any questions?
Any questions?
Do you know if there is any signs of inbreeding depression in the wild or captive population?
Not that we can see. So there's no obvious albinoism or anything like that going on. We do know that the habitats that are suitable are actually quite pressured. So there's a lot of fighting going on between the groups. These salmons have very complicated social structures. So there's usually only one breeding pair, and any mature female usually gets pushed out of the group, especially if she's showing signs that she is has the hormonal balance to get pregnant, as well as males tend to leave the group in order to be able to become the new dominant male. So there's a lot of switching going on. So it's hard to know if these reserves look like one huge family or if there is actually any genetic distinction between them. Yeah.
I recall you were also looking into their microbiome.
Yes. Yeah, that's another part of this project, is we do want to look into the microbiome of captive versus wild. However, our collaborators forgot to sample the fecal, so we currently only have captive. So we don't know how far we're going with that project anymore. It depends if they remember to collect it this next time around and if we can get it in time. Yeah, a bit of a bummer. Yes, Anna?
How old is the oldest historical sample that you have?
It's the oldest one I have is actually sort of undated. They know it's from the 1800s, but they don't know exactly when from the 1800s. But most of them are more recent. Most of them are in the 40s and 20s and 60s.
They're collected in museums as curiosities, or what's the context of having a skeleton of one of these guys?
Yeah, I'm not quite sure actually, to be honest. I did notice looking through a lot of museums, most of them started getting collected in the 50s. So I think it was this kind of random interests that kind of poked out. Maybe that was when they were interested in this area in Colombia or the rainforest, or who knows. But it does seem like in many different museums it was around that era or later. Yes.
How many individuals right now is in captive situations, and also are they like distributing different zoos, or like they're like more being together in one place?
All right, I'm going to answer. Tell me if I misunderstood. But the European zoos have about, I believe, over 7,000 individuals. The North American Zoo is about 2,000 to 3,000 individuals. So there's quite large populations there. Those are also the two largest, and then it drastically decreases after that. For example, the South American zoos, I think, only have like 100 and something, 200. Sorry, what was the second part of your question?
Yes, I don't understand concept of like the European zoos. Are this like the integrated facility, or like they are zoos?
Yes, so all the zoos in Europe have an option to join this region, which is known as the European Association for Zoo and Aquariums. And if they are part of that, then they join the breeding program. So they switch in between those zoos, but there's quite high level of, animal care and many other requirements they have to follow in order to be part of that. And it's to create a better situation for the animals, as well as trying to make a better breeding program. So it's not just, cousins and parents and offsprings breeding going on in the same zoo. So they do get transported around quite a bit. Same in North American zoos. They have also the AZA, which is their version, and then the same with South America. So they do get switched around. Yeah.
For the translocation between research, do you take in consideration the differentiation between populations or the phylogeny or something like that?
Yeah, so what we're hoping to look at is just first the overall genetic diversity, and then we're going to go in and look deeper. And the translocations are pretty much only going to happen if we can see a lot of inbreeding, or if it really is just one giant population that doesn't have a lot of diversity in it, then maybe we'll take some from the other reserve, but maybe both reserves look similar and then there's no point. So it really depends on what we find.
I have a technical question. So I've previously been faced with this lot of samples in my budget challenge, but I jumped on that very famous red seed train.
Yeah.
So what are the advantages with Mobisec, since I know it's new? Let's talk about it. Because transposable element recognition size sounds like something that's mobile within the genome. So does the same primer hit the same location in the genome in different individuals, or?
So for the most part, we try to choose a bit of a conserved T, where then they are mostly in the same area. I mean, there's obviously new jumpage that can happen.
Yeah.
So you have to be wise in which TE you choose, depending on your questions. But the idea behind choosing the transposable elements was that these areas are not being directly selected on. And so they could give us better information in that sense, instead of something that's causing a SNP because of climate or something like that. So it's to get a better idea of like what is the more baseline instead of what's being adapted, if that makes sense.
But I mean, 90% of rat-derived SNPs are also neutral.
Yeah. And I think it is very similar to that. Oh, Anas is probably going to add on this.
Yeah, the main difference is the targeted approach. Let's say if you have a complete sample, let's say it's just like this, and then you get the microbe, you get everything out. This one is designed primarily to target the organisms we want and get selected.
So for samples where you have
originally, so that maybe it's all samples. But we wanted to target the scat samples, scat samples and one track what individual did have, did deliver this scat and did have this viral, then you could use this targeting.
Okay.
The genome of the
So it's very specific to your target species.
yeah. That's the difference between the shotgun and the
Yeah.
No, it's good.
Makes sense.
I mean, by the sound of it, then you also need a reference, that to even start doing more research as opposed to RATSEC that you don't.
I mean, you do, like, we don't have a reference genus for this animal, so you can have someone that's closely related. So we chose one of the sister taxa. So as long as you know kind of that it's not too derived from them, then it seems to be working just fine.
I have one quick question related to what you said of the conservation when you do the design of the primers.
Mhm.
I guess at that point you also try to avoid highly duplicated regions because also elements, I'm assuming, they can also be highly repetitive and duplicated across the genome. Right? Is it part of the design of when you decide the primers to do single copy genes?
You mean like the tea itself isn't duplicated? Okay, I was just like, we want it throughout the Alice, I actually don't know this answer, do you?
In general, we are trying to look at the evolutionary history of the team, but of course, you never really know, right? Because some of the things with genomes today is that most of the mobile elements are not. To some extent, we know how much of the genome is mobile element, but not necessarily exactly where they are. Right? So it is a challenge on one side.
Yeah. Because there's also a question of how good that reference genome is, right? Because a duplicated region like that can be very hard to put in correctly.
Yeah, how many genomes you have in your alignment?
Yeah, exactly.
And it's because all of the wilds individuals resemble the scat.
No, the wild we have hair.
Okay.
We wanted scat as well, so we could do microbiome stuff, but they forgot to collect